Welcome back to MedTide Academy. In this video we will be walking through the process of glycolysis. By way of introduction, glucose is a type of sugar that serves as a preferred source of energy for most body tissues. Glucose is a complex molecule containing 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms, and it stores energy in its chemical bonds. However, body cells cannot utilize this energy directly from glucose. So to access this energy, the body needs to break down these chemical bonds through a series of chemical reactions called glycolysis, in order to release energy that can be utilized by the body cells. Glycolysis, therefore, is defined as the metabolic pathway in which glucose is broken down into pyruvate, producing a small amount of energy in form of ATP and NADH. Glycolysis takes place in the cytoplasm of cells, meaning that glucose will need to be transported from the bloodstream into the cells of the body. The molecules of glucose, however, are too large to pass through the cell membrane through diffusion. Therefore, they require specialized transporters in order for them to be transported into the cell. These transporters are called the glucose transporters, or GLUT for short. There are 4 major types of glucose transporters, and they are found in many different tissues of the body. They include GLUT1, GLUT2, GLUT3, and GLUT4. There is a mnemonics for you to remember the locations of these glucose transporters. This mnemonics is: big fat bullies kick, small little pancreas, producing nervous kids and mad fathers. So where do you find each of these transporters? For GLUT1, the B stands for blood, specifically the red blood cell. The F stands for fetus, while the B in bullies stands for the blood-brain barrier. For GLUT2, the K in kicks stands for the kidney, S in small represents the small intestine, the L in little stands for the liver, while P in pancakes represents the pancreas. For GLUT3, P stands for the placenta, N represents the neurons, while the K in kids stands for the kidney, again. The kidney has 2 types of glucose transporters, and this is because there is a multitude of cell types in the kidney, which require different types of transporters. Finally, for GLUT4, M in MAD represents muscles, while the F in fathers represents fats or adipose tissues. So these are where you can find the different types of glucose transporters. Also note that, out of all these glucose transporters mentioned, only GLUT4 is insulin dependent, so its efficiency in transporting glucose depends on the presence of insulin. Now let's discuss glycolysis proper. Glycolysis occurs in 10 steps, and these steps are divided into 2 phases. The first phase, which represents steps 1 to 5, is known as the investment phase, while the second phase, which occurs through steps 6 to 10, is known as the payoff phase. In the first phase, the cell invests or consumes energy, while the second phase produces energy, but more energy than what was consumed. In the first phase, the cell puts in 2 molecules of ATP to get the glycolysis going, but in the second phase, the cell gains 4 molecules of ATP. In addition, it also gains 2 molecules of NADH. Let's go over the 10 steps of glycolysis. Now, when glucose gets into the cell through the glucose transporter, it also has the tendency of leaving the cell through the same transporter, and this is because the glucose transporter is bidirectional. So to prevent this, the glucose molecule gets phosphorylated. Phosphorylation is simply the biochemical process in which a phosphate group is added to the glucose molecule. So in step 1, a phosphate group gets attached to the glucose on its 6th carbon atom to become glucose 6-phosphate. The phosphate group is gotten by the splitting off of 1 molecule of phosphate out of the 3-phosphate group of ATP, converting it from ATP to ADP. This step is catalyzed by hexokinase or glucokinase, depending on the cell where this is happening. Hexokinase is found in most tissues of the body, but primarily in muscle tissues, while glucokinase is found in the liver. It is also important to note that this kinase reaction is irreversible. In step 2, glucose 6-phosphate gets isomerized to fructose 6-phosphate. This simply means that the carbon atoms in glucose 6-phosphate got rearranged to form fructose 6-phosphate. This reaction is reversible, so fructose 6-phosphate can get reconverted to glucose 6-phosphate and vice versa. This reaction is also catalyzed by the enzyme phosphohexose isomerase. In step 3, fructose 6-phosphate gets further phosphorylated by the addition of another phosphate group to its first carbon atom, forming fructose 1,6-bisphosphate. Bi means that there are 2 phosphate group in the compound. But bis is used here because the phosphate groups in this compound are far apart. This reaction is catalyzed by the enzyme phosphofructokinase, PFK for short. And as usual, the phosphate group added to fructose 6-phosphate is derived from another molecule of ATP, which then gets converted to ADP. Step 3 is also an irreversible reaction. Moving on to step 4, fructose 1,6-bisphosphate, which has 6 carbon atoms, is cleaved into 2 molecules of 3 carbon atoms each. These molecules are dihydroxyacetone, DHAP for short, and glyceraldehyde 3-phosphate. This cleavage is catalyzed by the enzyme aldolase, and it is reversible. However, only glyceraldehyde 3-phosphate proceeds to step 5. DHAP, on the other hand, cannot proceed directly. Therefore, DHAP has to get isomerized to glyceraldehyde 3-phosphate first. This isomerization is catalyzed by the enzyme triose phosphate isomerase. This reaction is also reversible. Thus, at the end of step 4, we have 2 molecules of glyceraldehyde 3-phosphate. Moving on to step 5, a molecule of glyceraldehyde 3-phosphate is dehydrogenated and phosphorylated to 1,3-bisphosphoglycerate with the help of NAD+. In this reaction, NAD+ steals hydrogen from glyceraldehyde 3-phosphate, thus the term dehydrogenated, so it gets reduced to NADH plus a hydrogen ion. The phosphate group added to the first carbon atom to form 1,3-bisphosphoglycerate does not come from ATP this time. It comes from inorganic phosphate, that is PI, that are freely available in the cytosol. The dehydrogenation and phosphorylation that occurred in this reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. This reaction is also reversible. Step 6 is the start of the payoff phase. In step 6, the phosphate group on the first carbon atom of 1,3-bisphosphoglycerate is cleaved off to form 3-phosphoglycerate. The phosphate that was lost is transferred to ADP, which was formed in the previous steps, converting it to ATP. This reaction is catalyzed by the enzyme bisphosphoglycerate kinase, and it is reversible. In step 7, 3-phosphoglycerate is rearranged to form 2-phosphoglycerate. Essentially, the phosphate group was shifted from the third to the second carbon atom. This rearrangement is catalyzed by phosphoglyceromutase, and it is also reversible. Moving on. In step 8, 2-phosphoglycerate is converted to an enol, specifically phosphenol pyruvate, PEP for short. An enol is a type of organic compound that contains a carbon-carbon double bond, as we can see here, and a hydroxyl group. Comparing this to our PEP structure, we can see the carbon-carbon double bond right here. However, the hydroxyl group is replaced by a phosphate group. Thus, we can say that PEP is an enol phosphate. The conversion of 2-phosphoglycerate to PEP is catalyzed by the enzyme enolase, and this reaction is reversible. In step 9, phosphenol pyruvate is dephosphorylated to pyruvate by the enzyme pyruvate kinase. This simply means that the phosphate group was removed from phosphenol pyruvate. The phosphate was added to another molecule of ADP, also formed from the previous steps, converting it to ATP. This reaction is irreversible. Some authorities may argue that step 9 marks the end of the glycolytic pathway. However, in anaerobic conditions, pyruvate is reduced to lactase by lactase dehydrogenase. But in aerobic condition, pyruvate enters the citric acid cycle.